Okay, hello everyone. I'm Ahmad from University of Arkansas for Medical Sciences, and the title of this presentation is Machine Learning Powered Metabolite Identification in R and Automated Workflow for Identifying Metabolomics Dark Matter. But because of the dark matter, I just would like to start this presentation with this animation which simulated the formation of the dark matter in the whole universe from the beginning to now. So all of the dark and gray spaces show the unknown material in the whole universe, and only the bright spots shows the known part. Only 2% is just known according to estimations. And similar to the cosmology and the real world, also in the world of chemistry, most of the molecules and the small molecules in the chemistry are also unknown, and the number of unknown molecules are estimated to be very huge. 10 to power of 20 to 60 small molecules are still unknown, and only a very tiny part is known or measured, and even from the measured or measurable compounds or molecules, only a small part is identified. So identification is very high level characterization of the molecules, which is done by using very accurate and high resolution instruments like mass spectrometry. And the rate of the identification is very slow. Only about 1,600 molecules per year are identified per year. So compared to the whole unknown molecules in the universe, which is huge, still identification is very slow, and we need advanced tool for filling the gap in between them. So the idea is just using machine learning methods in order to improve the identification of unknown molecules for different purposes in metabolomics and in omics studies specifically, because we want to find the biomarkers for the diseases, following the pathway of the diseases, and even curing the diseases by using these molecules in drug discovery projects. So we need identification more and more. So as I mentioned, different instruments like mass spectrometry or gas chromatography, or even the combination of them, are used for identification purposes. I just brought a schematic view of the liquid chromatography mass spectrometry for identification of complex, for example, human fluids like blood. So at the beginning, the blood is injected into the column. So and then the molecules are separated based on their physical properties like lipophilicity or size, and then after they separated, they will imported into the mass spectrometer. They ionized and then they fragmented, and at the end we will have a detector at the end for detecting those fragments, and these fragments are shown here so as a signal, and this is called mass spectrum, and this is like a fingerprint of the molecule. And for the identification, we usually compare these fingerprints or these mass spectrum to the databases of the mass spectra for the known molecules, and as long as they would be similar together, so we can conclude that this compound is identified to one of those compounds inside the database. But the limitation is that the databases of the mass spectra are limited, and the number of known molecules in them, they are not more than 1 million molecules. So but the idea is that the number of unknown molecules is hugely large, so we do not have the mass spectrum for them. So so here is the place that machine learning methods can help for improving the identification. I also need to add that the data collected from mass spectra are very complex. I just showed the mass spectrum here, but it also has other dimensions like retention time and other properties, or even more mass spectral space. So data can be 3-way or 4-way. So, but here in this presentation, we only focus on mass spectra. So as I mentioned, the identification procedure includes comparing the acquired mass spectra from mass instrument to the databases. And as long as they match very precisely, we can call them level 1 identification. or if they deviate a little bit, we call them level 2, and still level 3 is acceptable, but beyond that, we call them dark matter or unknown molecule in metabolomics studies. So we can use different similarity measures for this purpose. For one of the most used methods is Tanimoto similarity coefficient, but it can different similarity measures can be used. So as I mentioned, the identification is very important in the multi-omics or metabolomics projects. We have different levels of information in our body hidden in the genes, in RNA, in proteins, in the metabolites. So we need the identification of these molecules in our body for better understanding of the diseases and the pathway of the diseases, biomarker discovery, drug discovery, and so in the metabolomics area as well, the science of knowing the metabolites in the body, we need the identification. So, but the problem is that the current mass spectral libraries have limited metabolite coverage, and less than 10% of metabolites have reference mass spectra, and in each metabolomics experiment, only about 2% of spectra can be annotated. The problem is small database coverage and low annotation rate, and these can be achieved using machine learning methods. I schematically presented here the main problem. The main problem is presented here schematically. So when we acquire the mass spectra from the instrument, some of them can be annotated to the database, some of them cannot. So those annotated, they are identified molecules, and those cannot be annotated, they are unknown compounds. So the idea is using the information here for relating the MS spectra to the structure of the molecules, because we know the structure of the molecules and we have the spectra, and we can connect them together by using machine learning methods, and then using those machine learning methods for identifying those unknown molecules. So here is the whole idea of the project. So we collect the MS spectra, train their machine learning models to the structures, and use those models for identification of unknown molecules. So just very simply saying that we are going to make some mathematical functions between the structure and mass spectra of the molecules, and then using those models for prediction of the unknowns. This also shows the whole picture of the work schematically. On the right top left, we have the MS spectra of all of our known molecules in a particular metabolomics study. We know the structure of those molecules. Because we know the structure, we can code the structure into the fingerprints. So all of those fingerprints on the right, they are known. We can calculate them. For example, if a particular molecule has a hydroxyl group, so those binary representations can show that. For example, one of those bits can represent a hydroxyl group in a molecule, and then for all of the substructures in the molecule, we will have all of those 0 and 1 binary representations, and then we can train the models between these two, I mean these fingerprints and these MS spectra. So as long as we have all of these for all of known molecules, and we have all of these fingerprints as well, we can train random forest models or any kind of models here to connect these two spaces together. And then after we train the models, we can predict the unknown molecules. So here is, because we do not know the structure of unknown molecule, we only have the mass spectrum. So we train this model. So we, as an input to this model, and this is the mass spectrum of unknown molecule, and then we can predict the fingerprint, I mean the molecular fingerprint of that unknown molecule. And then after, for identification of this unknown molecule, we need to search in a database, but these databases should be the databases of fingerprints, not the MS spectra, because we here have the fingerprint of these molecules. So we can create the fingerprint of all of the databases that we have, because it is simple and it's only a calculation. So it does not have the disadvantages of the MS spectral libraries, because as MS spectral libraries, they need to be acquired experimentally, but we do not have the experimental limitation here. For example, for HMDB, which is a Human Metabolome Database, we have about 200,000 molecules in that. We can calculate these fingerprints for all of the HMDB, or even PubChem, which is the most largest database of known molecules and includes 100 million molecules. We can calculate those fingerprints in this way, and then we can search inside in these databases easily, and then we can sort the number of candidates based on different similarity measures, and then we can identify an unknown molecule here. So the whole idea is that going beyond the MS spectral space, which had a lot of experimental limitations, into the fingerprint space, and then do everything and all of the identifications in the fingerprint space, and then we can identify the molecules. So the procedure had a lot of details for every metabolomics experiment that we do in the lab. We have a set of known molecules and a lot of unknown molecules. We can train the models by using the known molecules and use those models for prediction of the unknown molecules. The procedure includes data cleaning, organization, fingerprint calculation, and descriptor calculation, training the models, and then using those models for prediction of fingerprints, prediction of descriptors, and other properties like retention time. Finally, searching in database, HMDB, and sorting the molecules based on Tanimoto or other similarity coefficients. We also confined and funnelized the number of candidates more and more by using some of the estimated molecular properties like log-P, which shows the lipophilicity of the molecule and topological surface area and retention time, and etc. So in order to reduce the number of candidates more and more and increasing the accuracy of the identification. So I just show here the results as an example. For a particular plasma metabolomics study, we had 3,000 molecules in total, and 2,000 of them were non-identified and 1,000 identified. We trained the models, random forest models, by using those identified spectra in order to illuminate these non-identified molecules. So the whole procedure is summarized here. This is the compound discoverer, which is the software of the instrument. We extracted mass spectra into a small library of named mzVault. So from the mzVault, we went to SQL database, and then from SQL, we imported data into R. And by using RSQLite package, we handled that data. And by using SF package, we converted all of those bin structured data into numeric values. And then finally, we had the data matrix of all of the identified and non-identified molecules. We have the spectra. The dimension of this data is about 3,000 rows as molecules, and 30,000 columns. And the dimension is very high because the instrument is very high resolution. So every column shows a single MZ value acquired from the instrument. So because the dimension is very high, we need some dimension reduction methods. But the whole idea is just to connect this data matrix into the data matrix of the molecular fingerprints for the identification procedure. And so for reducing the dimension of the data, we used the sparse-PCA method because the data was very sparse, and also the usual PCA didn't work very well. And we also use RT-SNP package for another nonlinear representation as well. So this shows that 99% of the total variance of the data is described by 25 PCs. So we do not need to work with 30,000 dimensions. Only 25 PCs work as well. And these are the score plots of the PCA and T-SNP, which showing that identified and non-identified molecules are very overlapping. So it intensifies the hypothesis that we can use the MS spectra of the identified molecules for prediction of the non-identified molecules. So the data existing in there, and we can use identified for prediction of the non-identified. So color coding the PCA and t-SNP plots by using some substructure information in the molecules shows some repetitive patterns. So intensifies the hypothesis that we can use these models for prediction. So we train 166 random forest models for prediction of the 166 fingerprints, and then we use these fingerprints for searching inside HMDB or PubChem. And this is the overall accuracy of those 166 random forest models. And some of them were not very accurate, but in average, they were good. So it means that we can use these models for prediction purposes. And after searching inside HMDB, we realized that we still have a lot of number of candidates. So we needed to reduce the number of candidates. So here it shows the whole space of the Human Metabolome Database by using two representative physical properties like topological surface area, TPSA, and log-P. So it shows that if we confine the search space to some restricted area and using those filters, we can still reduce the number of candidates a lot. So we also had another model for other properties like retention time as other filters for reducing the number of candidates as well. So we finally defined a general scoring function, which is a combination of all of those filters. And we defined, and this is between 0 and 1, and shows the overall similarity of the molecule in the whole search space. So here is an example, which is this compound is unknown. We do not know what it is, and we only have the MS spectra. And then we predicted the fingerprints by using those 166 random forest models, and then we search inside HMDB. So the number of retrieved candidates are more than 1,000 molecules with the Tanimoto coefficient of more than 0.8. And we reduced those 1,000 molecules by using the filter of log-P into a smaller number, 891 hits. It also reduced more by using TPSA filter to 565. And finally, we implemented the retention time filter, and the number of hits reduced to 360. And we sorted all of those 360 by using the general scoring function that we defined. And then this is the top sorted molecules. All of them were very similar to each other. So it means that the developed workflow can find very similar number of molecules and converge to very small subset of molecules for the identification of a very unknown molecule that we didn't know that what it is. So these are all top marked molecules and suggestions for that unknown molecule. And finally, we did it for all of those 2,000 unknown molecules. And with the Tanimoto coefficient of more than 0.7, we could conclude that for at least 1,000 of those 2,000 molecules, a little bit more than 1,000, we could at least find one molecule in HMDB, which represents a candidate for that unknown molecule. So it means that at least we could achieve 57% improvement for the identification. For external validation, we used because we developed the models by using plasma. We use urine metabolomics data set for external validation. And for the level 1 molecules, we had 29 molecules, and all of them were inside the applicability domain of the models. This is an example of 4-oxoL-proline. This because it's external validation, we knew that what was that molecule. So this is the MS spectra of that. This is the predicted fingerprint by using our models, and this is the real fingerprint. So they were very similar. So, and this is the retrieved Tanimoto coefficients for searching inside HMDB, and it shows that the molecule came at the top list. So among 2,000 molecules, among 200,000 molecules, we could find that molecule at the top list. So other examples as well, we could find the molecules at the top, and some of them at the bottom list. So this is the overall AUC value for finding those molecules inside the ChemDB for level 1, and for level 2 we could achieve 0.73, which is quite acceptable. So we can conclude that for the challenge of the dark matter, metabolomic dark matter, we could develop an innovative workflow in R that can predict molecular fingerprint descriptors and retention time, and then combining them as a filter for increasing the accuracy of the identified molecules. And we also tested this method using external test sets, and we achieved still acceptable AUC values. I would like to thank Professor Colin Kay, Dr. Brian Piccolo, Dr. Rennie Lan, and Dr. Sari Chamtapalli, and Dr. Haley Astris from University of Arkansas for Medical Sciences and Arkansas Children Nutrition Center, and UAMS, ACH, and USDA for financial support of this project. Thank you.